Hello everyone and welcome to a new session on Aloasis Academia. On today's session we will focus on learning everything there is to know about eukaryotic cells ultrastructure. And here we refer to the internal structure of the cell that cannot be seen under light microscope, but you would need to use electronic microscope to be able to see these ultrastructures, these ultra small things within the cell. So we will cover their structure, their function, and how they are important to the cell. We will focus today on animal cell. In the part 2 of this video, we will go through plant cell and all the ultrastructure that will cover that. Don't forget that we have a revision booklet in my online website. In that booklet, it's going to be specifically on all the questions that have been asked on this topic for the past 15 years, and then give you all the keywords that you need to learn to specifically being able to hit all the marks for this topic specifically. Okay, double check it. You will benefit from it. Good. So let's start with having a look at a eukaryotic cell. So that's what a eukaryotic cell would look like. Okay, it has, as we see, it has a lot of stuff inside it, and we will go step by step and point by point to understand each of these internal structures that found within the cell. So this boundary here is the cell membrane that surround each cell and separate the inside of the cell from the outside. Okay, so that's the cell membrane, and these all the different internal structures we will learn today. So a eukaryotic cell has what we call a nucleus. So a nucleus is this part of the cell. And again, we will cover in more details what a nucleus is and what it does it contain. But for now, I just need you to be able to identify it on a schematic like that. So this is the nucleus. And then we have the other specialized internal structures. What do I mean with specialized internal structures here? So each of these internal structures we call organelles. Okay, so each of them, they are called an organelle. And an organelle is a specialized structure that is found inside the cell and have a particular function. Okay, so each of these organelles inside the cell, it has a specific role and a specific function that is playing within the cell. And many of these organelles, they are membrane bound. And when we say membrane bound, we just mean that the organelle has a membrane of its own, separate to the cell membrane. So membrane bound organelle, it means an organelle that is surrounded by a membrane. Okay, so this is a membrane bound organelle, which means it has a membrane of its own. So what's the benefit of having organelles within a cell? So the first thing is that the cell, each cell would need to have different reactions, okay, and each different chemical reactions, and each chemical reaction would need different conditions. So for example, some chemical reactions, they would need a pH of 7, other chemical reactions, they would need a pH of 4, for example. So the cell cannot have both pH at the same time if the cell doesn't have these compartments or organelles, what we call organelles. So the first benefit of having organelles is allowing the cell to have separate reaction in different places. So they act as metabolic compartments. So each of these organelles will have a chemical reaction inside it that is different to the other and would have also different conditions, and that would allow the cell to undergo these, for example, two different chemical reactions inside two different organelles within the same cell. So that's one of the very most important benefits of having organelles within a cell, being able to do chemical reactions that occur efficiently under different conditions. So I need you to remember that different cell types have different functions. So for example, a muscle cell would have a different function to a lung cell to a neuron cell. Okay, so a brain cell to a lung cell to a muscle cell, all of these would have different functions within the body. Okay, and because they have different function, then each of these cells will have different numbers and proportions of their organelles. So for example, we will cover that in more details in a few minutes, but for example here, if a cell requires a lot of energy, like a muscle cell, for example, it requires a lot of energy for contraction and movement of the body. So they would require a specific organelle called mitochondria, which is this organelle here. So then muscle cells would have a large amount of mitochondria because they need a lot of energy compared to, for example, another cell that doesn't require as much usage of energy as muscle cells. So that's I need you to remember. Different cells will have different proportions and different numbers of these organelles. Not all the cells have the same number and the same type of organelles. No, they are a bit different in numbers and proportions. Great. So let's start with the first thing, which is the nucleus. So as we said, that this part of the cell is called the nucleus. A closer look to it, it looks something like that. So what does it have? So in a eukaryotic cell, the nucleus has or considered to be the major genetic information compartments. Okay, and I said deliberately the major here because it's not the only place that has DNA. We will understand later on what other organelles in the cell that could have DNA. But what you need to understand at this point is that the nucleus is the major genetic information compartment. So it has the vast majority of genetic information that the cell has. It will be found in the nucleus. And as we can see, the nucleus is surrounded by a membrane, and the membrane is made up of two membranes. So it's a double membrane. And that's a very important keyword to use in the exam. So the nuclear envelope, it's a double membrane. It has an outer membrane, as we can see. This side is the outer membrane, and then that side of the membrane that faces the inside of the nucleus is the inner membrane. Okay, so it's a double membrane, has an inner membrane and an outer membrane. And as we can see in this image here, there are these structures that are extending from the nucleus. And we call that the endoplasmic reticulum. We will cover it in more details in a few minutes, but for now what you need to understand and learn is that the endoplasmic reticulum is a continuous extension of the outer membrane of the nuclear envelope. So that side of the nuclear envelope, the outer membrane of the nuclear envelope, they will extend to form these structures, what we call the endoplasmic reticulum. You also need to understand that as you can see in the image here, the nuclear envelope is not completely sealed. It has these small holes in it. And the reason or the main function of these holes is to allow materials to move from inside the nucleus to the outside and vice versa. So all this area within the cell, the empty spaces within the cell, it's called the cytoplasm. Okay, so some stuff in the cytoplasm might need to get inside the nucleus, and that will happen by going through these nuclear pores. And sometimes some stuff inside the nucleus needs to pass through these pores into the cytoplasm of the cell. That's why these pores are very important. So as we said, these called the nuclear pores, and they allow relatively large molecules to pass through the nuclear envelope. And an example of these molecules is messenger RNA. To understand what's messenger RNA and the different types of RNA we have in a eukaryotic cell, I have a whole video on my channel. Have a look at that, watch it. You will learn a lot from it to understand how mRNAs are made and what other different RNAs we have in the eukaryotic cell. Also, inside the nucleus, we have a jelly-like material that forms inside the nucleus, and that's called the nucleoplasm. Okay, so within the cell itself, we have the cytoplasm, which is these free spaces, and within the nucleus, we have the nucleoplasm. So do not mix them up. Cytoplasm is that area here. Nucleoplasm is within the nucleus, and it's a jelly-like material. So the genetic materials that are inside the nucleus, they are mainly consist of linear DNA. You need to understand and remember the word linear DNA, because we will in a few slides we will get introduced to the circular DNA that we have in a eukaryotic cell. However, in the nucleus of a eukaryotic cell, we have linear DNA, and this linear DNA, you might find it associated or bound to proteins. Okay, So this combination of DNA and proteins we call chromatin. So chromatin is the DNA with its bound proteins. And during cell division, we could refer to that as chromosomes. So chromosomes, they are only referred to during cell division. Again, I'm not going to go into a lot of details here because I have a whole video on my channel talking about the differences between chromatin and chromosomes and how the DNA will be wrapped around these proteins to form chromatin. And I have a video on mitosis to explain the cell division and how chromosomes are formed. So please have a look at that to get the full picture at the end and to improve your understanding. The last thing within the nucleus is this area here. So that's the nucleolus, okay? So you need now to start learning these terms so you do not mix them up, okay? So that's the nucleus, the nuclear envelope, and that's the DNA. These scribbles, these are the DNA. This is the DNA, and then this circular area here, it's the nucleolus. And the main function of the nucleolus is to produce ribosomal RNA, and also it's involving in assembling ribosomes. So if you are wondering what's a ribosomal RNA, again, in the same video in which I explained mRNAs, you will find a full explanation of ribosomal RNA, what are they and how they are made. And for ribosomes here, we will cover them in a few minutes to understand what's a ribosome. But for at this stage here, just you need to learn that nucleolus is the place of producing ribosomal RNAs, and it's involved in assembling the ribosomes. So the nucleus has to perform two important jobs within eukaryotic cell. So the first job is it stores the genetic material, which is the DNA. So it keeps the DNA protected inside the nucleus, protected from whatever happens in the cytoplasm outside the nucleus. So everything is protected inside the nucleus. That's why it's important for storing the DNA and keeping it protected and safe. The second thing is it acts as a control center. You might ask yourself why it acts as a control center. And that's because it has the DNA. So the DNA is the main source from which the cell will start making mRNAs, and from that will start making proteins. And those proteins will be running the whole function of the cell. So that's why the nucleus is acting as the control center. And any mutations or any problems with the DNA, this will affect the proteins, and this might affect even the cell and might even lead to the cell killing itself or the cell become faulty, like what happens in cancer and things like that. So that's why it is literally the control center of the whole cell. And here we refer to the nucleus. The next organelle that we will have look at now is the endoplasmic reticulum. And as we mentioned before, it is that part which extends from the outer membrane of the nuclear envelope. Okay, so let's zoom in. So that's what an ER look like when we zoom in. So ER, as we can see, has a three-dimensional system. Okay, of sheets like membranes. So we can see the membrane of the ER. It's made up they are very similar to sheets, and they are in a three-dimensional way. Okay, and they extend through the cytoplasm, as we can see there. So they start from the outer membrane of the nuclear envelope, and then they extend through the cytoplasm, as we can see here. So ER membranes form a network of tubules and flattened sacs that they are called cisterna. Okay, so cisterna, they are just the network of tubules and flattened sacs that the ER membranes form. So you need to understand this word because that's very important, and it's one of the main keywords that you need to write when answering about endoplasmic reticulums. Through the ER, we can. So now we will go to the rough ER. So what does this word here? So I've written it, but I didn't explain what does rough ER mean. So the first type of ER, it's called the rough ER. And the rough ER, it's called rough because it has these small circles attached to the outer surface or to the outer membrane of the endoplasmic reticulum. And these small circles are called the ribosomes. Okay. The main function of the rough ER, and here you need to pay attention that we are referring to the rough ER. In a few minutes, we will learn about the other type of ER. But for rough endoplasmic reticulum, the main function is that it provides large surface area, okay, for synthesizing proteins and glycoproteins. I know that the word large surface area is one of the most commonly used phrases that's in mark schemes, and that the examiners always look for that word to get a mark, and the students they just write it. So if you just here write that rough ER provides a large surface area, that's not enough, and you will not get the mark. Okay, you always need to say has a large surface area for what. So here you need to have the full information written in the exam. So the rough ER provides a large surface area for synthesizing proteins and glycoproteins. The second function is that it provides a pathway for the transport of materials like proteins through the cell. So these are the two main functions. They are involved in the production and synthesis of proteins and glycoproteins, and they are also involved in the transportation of proteins through the cell. So when the proteins want to transport from one place to the other, they are involved in that process. The other type of endoplasmic reticulum, it's called the smooth endoplasmic reticulum. And as we can see, smooth endoplasmic reticulum, they lack ribosomes. That's why they are called smooth. They don't have ribosomes attached to their outer membrane. Okay, and even in the shape, they are more tubular in appearance. Okay, so they are not like sheet-like membranes or flattened sacs. So these ones, they are more tubular in appearance, and their main function is that they are involved in synthesizing, storing, and transporting lipids and carbohydrates, and that's the main difference. Okay, so when we were talking about rough endoplasmic reticulum, it was mainly focused on proteins, whether synthesizing proteins and glycoproteins or transporting them. When it comes to the smooth ER, its main function, it's involved in synthesizing, storing, and transporting carbohydrates and lipids. So please remember that cells that tend to produce and store large amount of proteins, lipids, and carbohydrates, they tend to have extensively big endoplasmic reticulum. And that's the same point we mentioned a few minutes ago when we said that different cell types will have different function, and therefore they would have different proportions and numbers of the organelles within the cell. And one example is liver cells here and many secretory cells. So these cells that need to secrete, for example, a lot of enzymes, different types of enzymes involved in digestion and things like that, they would have extensively big ER to be able to synthesize proteins, transport proteins through rough ER, and also being able to synthesize, store, and transport lipids and carbohydrates. So these are the main keywords and the main information that you need to learn about endoplasmic reticulum. Let's have a closer look now at ribosomes. So all we know now about ribosomes is that they are found attached to the rough endoplasmic reticulum. But what they do look like, what's their function, we will cover all that. So in a schematic, just to make it easy for you to understand the structure of a ribosome, they would look like something like that. So ribosomes are small structures that are present in all cells. So whether it's a neuron, a muscle cell, a brain cell, a lung cell, they all have ribosomes. And the reason that all cells in the body has ribosomes is because ribosomes are the site for protein synthesis. So since all cells, they require proteins to carry out specific functions and become specialized, then all cells, they would need ribosomes, because it's the place, ribosomes is the place where protein is synthesized. If you need more information about how proteins are synthesized in ribosomes, again, I have a whole video on that in my channel. Please have a look at that. But for now, I will just explain it just in terms of the structure. So each ribosomal subunit, they contain ribosomal RNA and a protein. So what do I mean with ribosomal subunit? I mean these two subunits. So each ribosome is made up of a large subunit and a small subunit. Okay? And each ribosomal subunit would contain ribosomal RNA and a protein. Everything regarding how ribosomal RNA and proteins are involved in with ribosomes to synthesize the proteins, they are all covered in a video I made on my channel. Please have a look at that. So in the cytoplasm of a eukaryotic cell, ribosomes are called ATS. So you don't need to understand what S means here. It's just for rate of sedimentation, and it's related to how fast the ribosomes will form a sediment or a pellet when they are spun. But that's a whole different story. You don't need to know about it. What you need to know is that in a eukaryotic cell, you have an ATS, what we call an ATS ribosomes. They are 25 nanometer in diameter. And why you need to learn and remember that eukaryotic cells have ATS ribosomes is that it's because there are another type of ribosomes. They are smaller ribosomes, and they are called 70S ribosomes, and those are found in prokaryotic cells. So that's one of the main differences between a eukaryotic cell and a prokaryotic cell, is that in a eukaryotic cell, in the cytoplasm of a eukaryotic cell, you will find an ATS ribosomes. In a prokaryotic cell, you will find 70S ribosomes. However, you can find 70S ribosomes also in the mitochondria and chloroplasts. Okay, so ATS, they are always in the cytoplasm. 70, either prokaryotic, mitochondria, or chloroplasts. So the ribosomes, they can either be free in the cytoplasm, they will be just moving around the cytoplasm, or as we just learned, they can be attached to the rough endoplasmic reticulum. So these are the two main locations for ribosomes. And because the cells require to produce a large amount of proteins to carry out its function, ribosomes can account up for 25% of the cell's dry mass. So they are really high in number within the cell because the cell needs to produce a lot of proteins, and ribosomes are the site of protein synthesis. So now we know that proteins need to be made and they need to be transported through the ER. However, they also need to undergo further modifications. Once the protein are synthesized, you might need to add some stuff to the protein to make it fully functional and so the cell can then carry out its role. And now that's where the next organelle function will be very, very important for modifying the proteins that are made by the ribosomes and the endoplasmic reticulum. Great. So the organelle that I was referring to is the Golgi apparatus. So if we have a look at the cell that we had a look at the beginning of this session, so where out of all these organelles is Golgi? So that's Golgi. Okay, that's the Golgi apparatus. So Golgi apparatus is found in almost all eukaryotic cells. So it's very similar to the idea of ribosomes, which are also found in all cells. Golgi apparatus also present in all eukaryotic cells. Structurally, they are very similar to the smooth ER. If you remember when we showed the schematic of smooth ER, it has these flattened sacs. However, they are smooth. They don't have these small circles that are attached to the outer membrane. So they don't look like the rough ER. They look like the smooth ER. The difference is that they look more compact compared to this smooth ER. Okay. It also consists of these flattened membrane sacs. They are called the cisternae. Okay. Again, if you have a look at this word, remember where did we see it last time? So I'm not going to say, go back in the video and try to recap. Where did we see it last time? The other thing that, one of the most important functions of Golgi is that once the proteins and lipids are produced by the ER, whether the rough or the smooth endoplasmic reticulum, once they are produced by the ER, they pass through Golgi in an organized sequence. So what do we mean that the protein and lipids will be produced by ER and then passed by Golgi? So let's have a look at this schematic. So we have here the endoplasmic reticulum. So we can see the rough endoplasmic reticulum, which has all these ribosomes bound to it. And then we have the smooth endoplasmic reticulum. So what happens is that we would have these small vesicles. Okay, they will come off the endoplasmic reticulum with some, for example, proteins inside them, and then they will fuse with Golgi apparatus. Okay, once they are inside Golgi apparatus, what happens is that Golgi modify these molecules. So when the molecules enters Golgi, so we have that protein, it moved into Golgi, and then the protein is inside Golgi. So what happens is that Golgi will modify the molecules. So for example, it can add carbohydrate groups to proteins, forming glycoproteins. So the protein that was made by the endoplasmic reticulum, once it enters Golgi, it turned into a glycoprotein because in Golgi, a carbohydrate group was added to the protein. Another function of Golgi is that it sorts molecule so they can be sent to the right destination. So some of these proteins that enters Golgi and then they are modified, for example, they become glycoproteins. Some of those might be needed to be sent to the cell membrane. Others might be sent to another organelle inside the cell. So once they come off Golgi here, some will go, for example, to the cell membrane. Others might go to other organelles within the cell. So Golgi plays a role in sorting the destination and sorting the proteins, so it makes sure that each protein and each molecule goes to the right destination. So these Golgi vesicle, as we can see, they pinch off from the ends of the cisternae. So once they are pinched off, now they are free and they are sorted to go to the right destination. The Golgi vesicle, they contain modified proteins, as we said, maybe modified by carbohydrates. Also, they contain modified lipids, and they could be transported, for example, to the cell membrane, and then these vesicle will fuse with the cell membrane and then will be secreted outside the cell, for example. Maybe other ones will be sent to another organelle or to do another function within the cell. So you need to understand that Golgi plays a major role in sorting these, in modifying the molecules and in sorting their transportation. So it makes sure that each molecule arrive to this destination, to the right destination. In the exam, one of the most keywords that you need to always relate it to Golgi is that the main function of Golgi is to modify, sort, package, and transport. So these are the four main functions that Golgi does within the cell. I hope that made it more clear with the schematic. Great, let's get to the next organelle, which is lysosomes. Before we cover that, please, if you find this video helpful, support the channel through liking, commenting, subscribing. This will help me a lot and encourage me to upload more videos and make more lessons. So please support it. Thank you. Okay, so let's have a look at what lysosomes are. So one question that might comes to mind. So if the cell has digestive enzymes, then why a cell that contains digestive enzyme doesn't simply digest itself? Because it has the digestive enzymes inside it, why doesn't it kill itself or digest itself? Here, where it comes the main role of lysosomes. So if we go back to this image, so we have the endoplasmic reticulum, rough with ribosomes bound to it, and then the smooth. So they will be sending some proteins to the Golgi apparatus, and then within Golgi, as we said, proteins will be modified, proteins/enzymes. So even enzymes, they are type of proteins. So they will be sent to Golgi apparatus, they will be modified there, and then, as we said, they will be coming off the cisternae. Once they come from the cisternae, they will form these Golgi vesicles. Okay, Golgi vesicles are different types. One of the types of Golgi vesicles, they are called lysosomes. So when exactly do we call a Golgi vesicle a lysosome? Lysosomes are small membrane-bound structures containing hydrolytic enzyme. So if these Golgi vesicles contain hydrolytic enzymes, then we will call them lysosomes. So as we said, lysosomes are membrane bound, which means they are surrounded by a membrane. So they are membrane bound structures containing hydrolytic enzymes. So hydrolytic enzymes means enzymes that break down components, different components, depending on what type of enzymes they are. And there are different types of enzymes. So some enzymes will be proteases, which are enzymes that break down proteins. Others will be lipases, which are enzymes that break down lipids. Also, there is lysozymes. So lysozymes are enzymes that hydrolyzes components of the bacterial cell walls, and we will see in a minute or so when their role becomes very important. Okay, but for now, just you need just to learn that a lysosome, it's a vesicle produced by Golgi. It has specific hydrolytic enzymes, proteases, lipases, or lysozymes. And do not confuse the word lysosome to the word lysozyme. So lysosome, here we refer to the Golgi vesicle or vesicle. The lysozyme, we refer to the hydrolytic enzymes that break bacterial cell walls. So please do not confuse both of them. So a single lysosome may contain different enzymes. So once we have this lysosome, it may contain different enzymes. So it wouldn't have just one type of enzyme. It depends on what type of vesicle is it, but it might have different enzymes. And then here comes to the question that we asked, why a cell containing digestive enzymes doesn't simply digest itself, is because of the lysosome membrane. So the lysosome membrane will make sure that the hydrolytic enzymes within the lysosome are kept away from the cytoplasm and the inside of the cell. So as we mentioned before, that this free space is the cytoplasm, and then all the hydrolytic enzyme that could break down the cell, they will be kept away from everything with the lysosome membrane. So what can a lysosome do? So let's have a look at the different roles and function of lysosomes. So the first thing that a lysosome can do is to digest foreign materials. What do we mean with that? So that happens in what we call a phagocytic cells. So what happens is, for example, bacteria will bind to a specific receptors on the cell membrane. Okay, what happens is that the phagocytic cell will engulf these bacteria and gets it down, forming this phagosome. So this is called a phagosome. A phagosome is just engulfment. It will engulf these bacteria and then bring them down. Okay, and this is gonna be part of the cell membrane, and this forms a phagosome. So now the phagosome would have the bacteria. So now we need to break down this bacteria. Here where it comes the role of the lysosome. So the lysosome within the cell will come and then fuse with the phagosome, forming the phagolysosome. And then all the hydrolytic enzymes within the lysosome will interact with the bacteria within the phagosome and kill the bacteria. And here, as we said, it comes handy the lysozymes, which are able to break down the bacterial cell wall and kill the bacteria. So that's the first role of a lysosome, is to digest foreign materials. A phagosome can also digest worn out organelles. So any organelles that are old within the cell, they are no longer functional. So the lysosome can fuse with that organelle, and then all the hydrolytic enzymes within the lysosome will be secreted into that organelle and then break down the whole organelle. Once the organelle is broken down, this is an important info, is that the useful chemicals will be reused by the cell. Okay, so nothing is wasted. So even if there is an old organelle, the lysosome will fuse with the old organelle, break it down, and then any useful chemicals will be reused by the cell. Another function of a lysosome is to be released outside the cell through a process called exocytosis. So for example, if this lysosome was formed and it has a specific enzymes that are needed to, for example, break down some food, okay? So this enzyme needs now to be secreted outside the cell. So what happens is that this lysosome, and as we said, Golgi will make sure that this lysosome will be directed to the right destination, which is the cell membrane. So this lysosome will go to the cell membrane, fuse with the cell membrane, and then secrete all the enzymes outside the cell. And that's through a process called exocytosis. The final role of lysosomes are performing autolysis. So autolysis is the process where a cell wants to kill itself, for example. Okay. So what happens is the lysosome, the membrane of the lysosome, will break itself, and this will allow the hydrolytic enzymes to be secreted inside the cell, and this will result in enzymatic breakdown of dead cells. Okay, so the cell will kill itself by the hydrolytic enzymes that are in the lysosome. So these are the main functional benefits and roles of lysosomes. Great, let's now cover the last organelle that we will learn about in this video, which is the mitochondria. So what is the mitochondria, what its structure, and what is the main function of a mitochondria? So as we can see here, the mitochondria is a rod-shaped organelle surrounded by two membranes. So the first membrane is the outer membrane, which is the one outside, okay? And then we have this inner membrane. And between the outer membrane and the inner membrane, there is the intermembrane space. So there is some space between. However, it has two membranes, the outer membrane and the inner membrane, okay? The inner membrane, as we can see, it's folded inwards, okay, to form more extensions, okay, and these folds and extensions, they are called cristia, okay. So that's different to the cisternae. So cisternae, it's when we covered what's in the Golgi apparatus in the endoplasmic reticulum. So those flattened sacs and folds, they are called the cisternae. However, in the mitochondria, we here refer to the cristae. Okay, so cristae, as we said, is the inner membrane being folded inwards to form these extensions. So what's the role of cristia? So cristia provides large surface area, and as we said, just saying that cristia provides large surface area doesn't get you the mark in the exam. So you need to say for what. So cristia provides large surface area for the attachments of enzymes and other proteins that are involved in respiration, because as we know that mitochondria, it involves and it plays a major role in respiration. That's why you need to understand that these cristia folds, they provide large surface area for the attachment of enzymes and proteins that are all involved in respiration. The remainder of the mitochondria, it's called the matrix. So this place here inside, it's called the matrix. Let's zoom in into the matrix. What does the matrix contain? So this area here within the inner membrane, it's the matrix, and it contains ribosomes, which we already know by now what are they. It contains circular DNA, and if you remember that in the nucleus of a eukaryotic cell, it has a linear DNA. However, in the mitochondria, we have circular DNA. And also another thing for ribosomes. So which ribosomes do we have here? Is it the 80S or the 70S? So maybe you need to revise a little bit what we said earlier and remember what ribosomes are there. So we know that the ribosomes can be found free in the cytoplasm. They can be attached to the rough endoplasmic reticulum, and also they can be found in the chloroplast or mitochondria in animal cells. So since this is an animal cell, it has a mitochondria. What ribosome is in there? Is it 80S or 70S? Also, we have granules, which are bound to the circular DNA. Also, we have ATP synthase particles. So we know that ATP is the main source of energy that the cell uses, and a mitochondria, is one of the main producers and has a main role in producing the large amount of ATP that the cell need. Other things that can be found in the matrix is other enzymes that are involved in respiration, also proteins and lipids. So all they can be found in the matrix here in the middle. So here is an important point. The fact that the mitochondria has a DNA of its own, although it's a circular DNA, but it's a DNA of its own, and a ribosomes, as we can see there, then this allows the mitochondria to produce some, not all, but some of their own proteins. Because as I mentioned in a different video about how proteins are made, so you have a DNA, the DNA will be transcribed into an mRNA, and then mRNA will be translated into the protein, and you would need a ribosome to do that. Again, the process is explained in way more details in a different video. Please have a look in my channel, you will find it there. However, but you need to understand this idea that the mitochondria can make some of their own proteins because they have ribosomes and DNA. Also, mitochondria is the main site of aerobic respiration, therefore have a major role in ATP production. That's why mitochondria are considered to be one of the main things that are important for producing large amount of ATP for the cell, because it's the main site for the aerobic respiration. Also, some cells, they might need more ATP than other cells. So for example, muscle cells, because for muscle contractions, they use a lot of ATP. So these type of cells, they would have more mitochondria than other cells, like for example, lung cells, for example. And as we said, this takes us to the very beginning of this video. The first concept we introduced is that different cell types will have different function, and therefore they would have different numbers and proportions of organelles. So if a cell needs a lot of ATP, then it would have a lot of mitochondria to produce a lot of ATP. And also, if we look at the Christia, we can see that more densely arranged Christia are associated with greater metabolic activity. So that's also an important point. More folds are associated with a greater metabolic activity. Why? Because increasing the folds will increase the surface area. For what? For attachment of enzymes and proteins, and that's why you will have a greater metabolic activity at the end. So that's an important point that you need to keep in mind. So always ask yourself, that's the main thing of this whole lesson today, is always ask yourself, can I make the connection between the structure of the, for example, organelle I'm studying and its function? Can I make this connection? If you can make it clearly, then you made the most out of this lesson. To wrap up, these are a summary of the most important functions of each of the organelles. Nucleus, it's important for storing genetic materials and controls cell activity. As we said, it acts as a center of control because of the DNA that it has and stores. Ribosomes, important for protein synthesis. Rough endoplasmic reticulum, important for protein and glycoprotein synthesis and transport. Smooth endoplasmic reticulum, important for lipid and carbohydrates synthesis, storage, and transport. Golgi apparatus, it's important for modifying, sorting, packaging, and transporting. Lysosomes are for controlled digestion and recycling any worn out materials inside the cell. And finally, mitochondria, it's for aerobic respiration and ATP production. I hope you found this lesson helpful. Stay tuned for the part 2, which we will cover the organelles that are more related to the plant cells. We'll go into more details on that and cover all the organelles. Thank you, and see you in the new video.